So today I'd like to talk to you guys about some of the molecular adaptations to life in very cold environments. And I'm going to specifically talk about a bacteria that we've isolated recently in my laboratory called Planococcus halocryophilus. So just to give a little bit of background, most of the work in my laboratory is done in the Arctic or with microbial habitats and or microbes that live in the Arctic. So there are a number of unique microbial habitats that we find there. They include hypersaline springs, permafrost, and cryptoendoliths, which you maybe can tell Jackie was having a hard time earlier, but you can see maybe the green layer there, which is basically made up of microbes. So these habitats are also pretty good terrestrial analogs for certain places in our solar system that are kind of cold, frozen, or salty. But they're also really good places for the study of cryophiles or extremophiles that like really, really cold environments. So just to situate you a little bit, this is us somewhere right around here. And if you enlarge this area up here, this is Ellesmere Island and Axel Heiberg Island. And a lot of the research that is done in my lab is done here. And Planococcus, the bacteria that I'm going to talk about, was actually isolated from this region here, Eureka in Nunavut. So Planococcus halocryophilus is really cool, really interesting, because it actually grows at the lowest temperature that we've ever recorded to date, which is minus 15 degrees Celsius. And it also tolerates up to 19% salt, which is very high. This is a scanning electron micrograph of Planococcus. So it's a diplococci, cocci meaning that it's round, and diplo meaning that it usually lives with another one of itself joined together. So most of the work that pretty much all the work that I'm going to present today is actually from an ex-postdoc in my laboratory. Her name is Nadia Mikichuk. And so she isolated this bacteria and then did a bunch of work looking at the molecular adaptations of this organism. So she looked at genomic traits. She basically sequenced the whole genome. She looked at she did transcriptomic analysis, which is basically looking at or comparing which genes are active in certain conditions compared to, which genes are active, for example, at room temperature compared to which genes are active at minus 15 degrees Celsius. But one of the first things that she did is she looked at the bacteria under the microscope. So these are scanning electron micrographs. And she compared what they look like at different temperatures. So this up here is 25 degrees Celsius, and this is what bacteria usually look like. This is what we see, and this is what Planococcus usually looks like at 25. When you look at it at minus 5, you can see that it starts to change a little bit. The surface looks a little bit more different. Not sure what's going on, but it looks a little strange. When you go to minus 15, it looks completely different. So something is happening to the surface of the bacteria at minus 15 degrees Celsius. Now, I wish I could tell you that we know exactly what's going on, that we know exactly what this is and the reason why it's there, but we don't. We're looking into that right now. Jen is actually looking into maybe what this is composed of and the reason why this envelope is there. There's a few different possibilities. It could be a mixture of different things. It could be proteins, lipids, maybe biofilm of some sort. It could be just residue from cell wall when the bacteria separates. Where the actual separation, when the bacteria replicates, where that happens, it could be just extra cell membrane that is just left there. But we don't really know right now exactly what's going on. So the next thing that Nadia did after she looked at it under the microscope is she sequenced the entire genome. And then she analyzed the genome for essentially adaptations that she could find for life in cold environments. And one of the things that she found is what we call genomic plasticity. And so it's this idea that organisms like Planococcus, organisms that live in really cold or stressful environments, have many copies of the same gene. So they basically have many versions of the same gene. And the reason why they do this is this basically allows them to survive in many different stressful conditions. So for example, in a warmer or a salt condition, you might use one version of a gene, whereas in another condition, like a colder condition, you'll use another version of a gene. So depending on which condition you're in, which stress you have, you're going to be using a different version of the same gene. And each version is a little bit different, and it allows you it allows the organism to use that same gene, but in these different environmental stressors. So you can see for Planococcus, for example, this is looking at choline betaine uptake. So this is looking at solute uptake and synthesis. And solute is really important for the bacteria in order to allow it to survive in really high salt conditions. So it accumulates these solutes, these little organic molecules inside the cell, and they actually allow it to survive 18, 19% salt and really high salt percentages. And so if you look at just these genes that are involved in this process, Planococcus has a lot of them and has many copies of these genes. So it's this idea then that depending on the environment it will find itself in, it can upregulate one version of the same gene, whichever one is necessary for the environment that it's in. So the other thing that she looked at was the idea of, or she looked at amino acid changes. So you have a gene sequence, and cold-adapted proteins will actually change. So essentially, if you take a gene, your gene codes for amino acids, which then make a protein. And which amino acids are in that protein can actually change. And so this is the idea of increasing protein flexibility. So if you look at these, like these two here, what she essentially found is most of the genes in Planococcus actually have less acetic and proline residues. So they're basically cold adapted because having less of these gives you increased protein flexibility. So a lot of these genes are essentially better adapted to functioning in a cold environment. Oops. So the next thing that she did was then transcriptomic analysis. So she basically looked at which genes were active at -15 and which genes were active at high salt and which genes were active at room temperature. And then she compared them to essentially get a better idea of which processes inside the cell were involved in high salt or -15 conditions. So the main thing that she found, or these are essentially two of the main things that she found, which is one, there's decreased expression across functional categories. And that just basically means that in general, the bacteria shuts down most processes that aren't necessary. So a lot of genes that aren't necessary, they're not going to be active. You don't want to waste energy having a lot of things active that you don't need to have. So most cellular functions are repressed, but she did find upregulation of a number of genes involved in cell wall modifications, which we can expect based on the pictures that we saw earlier. Amino acid metabolism, as well as oxidative stress response, which is normal when you're under stress, and changes in transcriptional regulation, or a lot of transcriptional regulation. So again, you can easily shut on and off genes that you need or don't need. So this is the last picture that I want to show you guys, which kind of looks really confusing and complicated. It's not really, well, it is sort of. This is basically a very It is, but it's basically a very brief overview of all of the different processes that are happening at any single time inside of a cell. And that's not even nearly close to everything that's happening inside of a cell. But in terms of Planococcus, what we want to highlight are basically the genome-encoded adaptations, cold adaptations, that Nadia found to be important. And so this is just generally all the main sort of categories of adaptations that she found in the genome. So she found adaptations in cold shock genes or cold shock proteins, which would make sense, compatible solute uptake, cell envelope modification, and things of that nature. But she did find that some processes were induced more either by high salt or more by cold. And so these are highlighted here. So things like lipid synthesis and turnover, amino acid turnover, oxidative stress, these seem to be more induced by the high salt conditions. And in the -15 conditions, she found, again, translation, transcription, and what I find really interesting, which are these hypothetical proteins. So these are essentially proteins that we've never characterized before and that actually might be really, really important for cold survival. But we just don't know what they do because we've never seen them before. And so I think that's one area of research in the future that would be really interesting, which is to look into what these proteins are and what's their role in cold survival, because they may be really interesting. So the current work that is going on in our laboratory involves looking at the composition of the -15 envelope, which I mentioned, which Jen is looking into. We're also doing some proteomics, looking at, so the next steps, we looked at genes, we looked at which genes are active and which aren't, and then we actually want to look at the proteins inside the cells and figure out what's actually going on in terms of the proteins, in terms of the molecular mechanisms inside the cell. And then also looking at compatible solutes, so figuring out how Planococcus is actually able to tolerate such high salt, what solutes it actually accumulates inside the cell for salt tolerance. And that's it.
So questions for Isabelle?
Have you thought of like overexpressing some of these proteins in like E. coli or something and seeing if they enable maybe cold adaptations?
You mean the hypothetical proteins? Or?
Well, well, you could hypothetical, but more safely, some of the ones that affect like the lipid membrane and stuff like that.
Oh yeah, for sure. There's
Just
so many things that you can do with this organism at this point.
Yeah.
And so we're just at the beginning of looking at this more, but there's a lot of cool stuff you can do, yeah.
It was my turn or online turn. Is your organism genetically tractable?
What do you mean by genetically
sorry, I mean,
tractable?
are you able to do genetics with your organism, like make mutants with it?
I'm sure you could.
Oh, okay, I was just wondering if your lab also makes mutants with the organism to investigate like the function of like the hypothetical proteins.
Oh yeah, no, we haven't done that yet, but we definitely could.
Okay,
yeah.
cool.
Yeah.
So I guess we'll take an online question.
Okay, this is from Batul. Her first question is, why is protein flexibility beneficial in cold environments?
So I guess this is sort of the idea that with the cold, you want to have proteins that are very flexible. It's easier to bind to your substrate if you're not so rigid, which is probably what's going to happen at lower temperatures. So something like proline creates kinks and rigidity in an amino acid. So you'd rather not have that so that you're more flexible. And when you have the cold, which tends to kind of want to slow you down, it essentially allows you to better bind to your substrate.
Okay, and our second one is, could you tell which proteins involved in cell wall membrane regulation et al. Are adapted?
Can you say that again?
So, could you tell which proteins involved in cell wall membrane regulation are adapted?
You mean cold adapted, or? I don't think we've looked into that specifically, but that's definitely something that we're going to look into in the future, because that's what we're working on right now.
Thanks, Isabelle.